it works. Okay, I'll leave you. Good evening, ladies and gentlemen. This is the gene revolution. Can you hear?
Yes.
Okay. I also give it a subtitle. It's the tale of two molecules. You're all familiar with the double helix, DNA. I'm going to talk a lot about genetic engineering tonight. But I'm also going to talk about the small molecule. The small molecule is glycine methylphosphonate. It's a chelating agent. It's the active ingredient of Roundup, glyphosate. And we'll spend some time with that. So this is what I'm going to talk about to start with, what GMOs are and how they're made, because most people don't really know and are always very curious. I'll make it very simple. And then I'm going to review the claims of the industry that genetically engineered crops reduce pesticides and have higher yields and are completely without effect on the environment and are perfectly safe to eat. I'm going to spend a good amount of time on that. So what are GMOs? Well, genetically engineered crops today are herbicide resistant. Over 90% of all engineered plants on the planet today are engineered to resist the herbicide Roundup. So really, the revolution, the gene revolution, is about selling a chemical, a herbicide. There's nothing else. Well, almost nothing else. Everything else is a distraction. So when you hear about golden rice, or drought resistant, or all these high in the clouds technologies that are going to come in the future, the reality today and for the last 15 to 20 years is this: herbicide tolerance. HT stands for herbicide tolerance. And the herbicide is Roundup, so you have Roundup Ready corn, Roundup Ready soybean, canola, cotton, sugar beet. 100% of sugar beet today are engineered. Wheat, potatoes, etc., etc. All the major crops have been engineered, alfalfa, have been engineered to tolerate, to resist the herbicide. There is a second technology, a second trait. It's called BT. It's to make the plants resistant to insects. And that's a much, much smaller technology. It's really 10 to 15% of the plants that are engineered have that trait. Mostly it's the herbicide. BT stands for Bacillus thuringiensis. It's the name of a bacteria that was discovered 100 years ago in Japan. Then it was forgotten. Then it was discovered again after World War II. It's a natural bacteria, and it kills caterpillars, it kills a lot of insects. It's a whole group, a whole family of bacteria that makes a protein that is extremely toxic. It's toxic. Some of them are toxic to some insects, some of them are toxic to some other insects, some of them are toxic we don't know what to. Many, many different kinds of proteins that are toxic. BT. And there is no denying the incredible success of the technology. Today, the curve here starts in 1996. This is when the first crops were commercialized, when the first genetically engineered herbicide-resistant crops were commercialized, corn and soy, 1996. Start from zero. And today, I think it goes to 2010, today we're in 2013, 90%, like I said, 90% of corn, soybean, etc., 100% of sugar beet have been are now grown by the farmers. This herbicide tolerance in the plant makes for a magic technology. It's really easy to market it for the company. You know, if you're a farmer or even a gardener, the weeds are a serious problem. Weed management is the most important problem in agriculture today. 80% of all the pesticides we have, all the pesticides we use in agriculture, 80% are herbicides. That's how important the weeds are. So imagine a company that comes and says, well, you know what, I have this magic herbicide. It's very safe, it's totally safe, it's innocuous. no effect on the environment, nothing, or on your health. And you don't need to worry about your weeds anymore. You just seed your crops, or you seed your vegetables in your garden, and then a week or two later, or a few weeks later, whenever you want, you apply the herbicide, and all the weeds will disappear. Guaranteed. It's a dream technology. For the farmers, it simplifies their life. They love it, and this is the proof. There's nothing like it. This is high-tech, and they absolutely love it. This is what GMOs are today. HT, herbicide tolerance, and BT, insect tolerance. I think I should have taken a minute to explain what BT is about. So you have this protein in the bacteria that's very toxic to the insects. If you're a genetic engineer, you find a gene for that protein in the bacteria. You put that gene into the plant, and the plant is now insecticidal because every cell of the plant is producing the protein. When the insect comes to feed on the plant, they die. The plant has become its own pesticide, and you don't need to spray insecticide. And that also the farmers like to. So, how were they created? Well, there's basically two technologies. The first one that was studied was when we realized that there's a normal bacteria, a common bacteria in the soil, which causes a crown gall disease. Maybe some of you know it. You can see it on the thimbleberry plant in the forest. You can see those galls on the plants. Some of them are caused by insect. Many of them are caused by this bacteria. The bacteria is called Agrobacterium tumefaciens because it causes those tumors. These are tumors, basically cancerous cells. And what the bacteria has learned to do, has evolved to do, is it to transfer its gene to the plant, to a plant cell, just like the bacteria do to each other. The bacteria transfer their genes to each other very easily. It's just like, you know, you just sit next to the other bacteria and you say, here, have some of my genes. And this bacteria, this particular species of bacteria, has learned to do that with a plant. And so it transfers some of its genes, the genes that are absolutely necessary. They're all on the small plasmid. The bacteria, all bacteria have a chromosome. They don't have a nucleus. They have a big chromosome, and then tiny mini chromosome called plasmids. And all the genetic information necessary for the gene transfer to the plant are on a plasmid called the TI plasmid, the tumor-inducing plasmid. And if you're a genetic engineer and you know how that works, all you need to do is to replace the genetic information on the TI plasmid with what you want, with the genetic information you want to transfer into the plant. And you replace that. And we have the tools for that. They're proteins, they're enzymes. Enzymes, cut and paste. You cut a piece of DNA, and then you paste another one instead. And then the bacteria didn't feel a thing, it doesn't know what happened. And the bacteria comes next to the plant, and it will transfer its genes to the plant cell as it would normally, and it has transfers the genes that you want. When the bacteria does it naturally to the plant, it transfers its own gene. It's parasitism, because really, the genetic information going into the plant cell is just to tell the plant cell, now you're going to do what I want. And the bacteria says, feed me. And the plant cell makes sugar that do not exist in the plant kingdom, for the bacteria. So now, as a genetic engineer, you've learned all that, and you've engineered the plant cell, and now the plant cell will do what you want. And it could be whatever you want. You could have put a fish gene, you could put a human gene, you could put a plant gene, it doesn't matter. Then the plant cell that has been genetically engineered will do exactly what you wanted. That's the first technology. The next one is called the gene gun. And the gene gun looks like a hairdryer. It's a gun. You're shooting millions of very tiny pellets of metal, platinum or gold or something like that, that you coat with, again, the same genes that you want in the plant. and you coat the pellets with the genes, and then you shoot millions of pellets into the plant tissue, into the leaf tissue, and somehow one or a few of those pellets are going to find, I mean the pellets go everywhere, it's really a shotgun, and one or a few of the pellets are going to find their way into the nucleus of a cell, and then the gene construct, the genes that you have coated the pellets with, are going to be integrated into one of the chromosomes of the plant cell. And now the plant cell is engineered. The genes are now in the chromosomes of the plant cell. It's engineered. You have engineered the plant. When you've done that, you need to, same thing with the agrobacterium technology, you need to see which cells have been engineered, because it's incredibly inefficient process. And so you've shot millions and millions of pellets, and you do the experiment, you know, you're gun happy, you shoot. And you need to disintegrate the tissue so you get single cells, you get rid of all the plant walls of the cells, the cell walls, and then you plate the cells onto a petri dish. Which ones have been engineered? 1 in a million, perhaps. So you've got 1 million cells, and you have to find the one that has been engineered. And to do that, you use antibiotic, because you knew you were going to use antibiotic. With the gene that you've coated the pellets with, you have placed a gene for antibiotic resistance. Bacteria, a lot of bacteria are antibiotic resistant these days. It's easy to find a gene for antibiotic resistance. So now you have the gene that you want, plus a gene for antibiotic resistance. You shoot that into the plant cell, they go into the chromosome, the cell is engineered. You put that on a petri dish with some antibiotic, and only the plant cells that have the antibiotic resistance genes will not be killed by the antibiotic. So all the cells die except one or a very few that have been engineered. And now you have a cell, a plant cell, could be corn, that is engineered. And using the proper dosages of quite a few plant hormones, and it's quite sophisticated, tissue culture, you can regenerate that plant cell you use as an embryonic cell, like a stem cell. And you regenerate a whole plant out of it. And then when you have a whole plant, you can multiply that plant and have hundreds of millions of acres of engineered plants. And this is what we have today. So that's the technology, made simple. Okay, so how do those crops perform? And do they stand up to the promises of the biotech industry? Well, the first claim of the biotech industry is that this technology was invented to reduce the use of pesticides. And so remember I said there is herbicide tolerance and insect resistance. And if you have a plant that is insect resistant, that when the insect come on the plant and eat the plant, they die instantly because the plant is toxic to the insect, you don't need to spray insecticide. And so yes, for quite a few years, this technology worked very well, and there was no need to spray insecticide. But it happened very quickly in a few years, what always has happened when we use insecticide technology for the last 40 years or 50 years, that the insects are adapting. They are becoming resistant. And the farmers have known that for 50 years. You use an insecticide, it's the new product today. It's wonderful, it kills everything. And a few years later, well, it doesn't work so well. Lots of species of insects are now resistant. And then soon, it doesn't work at all. Oh, that's okay, we've got the new technology, a new insecticide that works really well. And that also works for a few years. It's normal, it's biology. If you put enough of the chemical to kill a pesticide, to kill any biological organism, they will adapt. It's the same thing with antibiotic and bacteria. They will adapt. And that happened with this technology. The BT technology is becoming less and less, works less and less because the bugs are becoming resistant. So now the growers are told to spray insecticide on their engineered crops. So that's for the insecticide. For the herbicide, it's a very, very different story. The herbicide, right from the start, the curves went up sky high. The usage of Roundup, because that's the chemical, the usage of Roundup has increased 1,500% in the last 15 years. The curve here goes on into 2007, and it keeps going up and up and up, even faster, like a hockey stick, because now we have resistant weeds. We have super weeds, and there's a lot of them. There's over 40 species of weeds now that are resistant to the herbicide Roundup in North America. And so the growers are stuck with this failing technology. They have either to pull the weeds by hand when you've got, you know, thousands and thousands of acres, it's not really working very well, or use other chemicals. The weeds are infesting about half of the acreage in the US. And they're in Ontario, they're in Manitoba, they're in Saskatchewan, and they're in Alberta. And we don't have them, I have no report of them in BC yet. And there's no reason to think that they won't be here next year or the year after. Everybody knew, every scientist knew this was going to happen, and of course the industry knew very well this was going to happen. And so they've had many years to invent the next technology, the next chemical. So now instead of spraying Roundup, we are going to spray 2,4-D. 2,4-D was in the Vietnam War, Agent Orange. 2,4-D has a history of being somewhat toxic. Definitely causes Parkinson's disease. In Vietnam, the manufacturing process was such that there was dioxins with him. Dioxins are teratogenic. And I could say, I can assure you, that we won't be spraying dioxins on the North American continent. So, not really, there is no reduction in pesticide. Actually, quite the opposite, which makes perfect sense for a chemical company. It would make no sense for a chemical company to invent a technology that would reduce their sales. The next claim of the industry is that this technology increases yield. Now, yield is a multi-trait. It would take like hundreds of genes to change the yield of a plant, particularly to increase the yield of a plant. You can't increase the yield of a plant just by changing a gene or two. That is absolute nonsense. So, of course, this has not happened at all. Actually, quite the opposite. This correlation curve is difficult to read, so don't bother. This basically tells you that the average yield of corn in the last 15 years in Europe was lower 15 years ago than it was in North America. And 15 years later, the yields of corn in Europe are higher than they are in North America today. In North America, we use genetic engineering. Over 90% of the corn is engineered. In Europe, 0% of the corn is engineered. They do not use this technology, and they get higher yields. The technology does not increase yield. This document was published a few years ago. It showed the results of experiments that were done in US universities, Illinois, Kansas, Iowa State University, showing basically data that showed very clearly that what they call this a yield drag. The engineering process of the plant, particularly in soybean, is actually costly, physiologically speaking, to the plant, and the plant yields less. And basically the same with corn. And the growers know that, and the industry knows that. But because the weed management is so simplified and so safe, they are willing to go with less yield and less revenue because of that. It's convenience. It's the convenience effect. But definitely no increase in yield. That's a false claim. The next claim is that this technology has no impact on the environment. And I'm just going to discuss two kinds of impact that I see. The first one is gene flow. You have engineered plants, and they have different genes. They have at least a different gene for herbicide tolerance and a different gene for antibiotic resistance. And those genes are going to fly in the pollen of those plants. So that if you are organic growers next to me and I'm having this huge 1,000-acre field of corn that's engineered, you are going to get showers of pollen, and much of your grain in your field is now going to be having those genes. It's gene flow. You have been contaminated. And if you're organic and you want to sell your product as organic, sometimes you will have to show that you are organic, and I'm sorry, you've been contaminated, it's not organic anymore, you've lost your crop. It's good for the pigs. Or you want to export it to Asia or to Europe, and it's not organic anymore. No, they don't want the technology there. And if you want to use your seeds next year to replant, you will have engineered plants, many of them, lots of them in your field. Now either you're unlucky and Monsanto will come and sue you for stealing their technology, or else you're smart and you'll sue Monsanto and ask them to come and remove those plants that belong to them. But this is a serious problem for conventional and organic growers. Many of them are really upset with the industry about that. And in Canada, well, we've lost markets. We've lost the European market for canola because of contamination. We've lost the flax market because of contamination. And a lot of people are very worried about losing markets for alfalfa, because alfalfa is coming. And alfalfa is a very important crop for the organic growers and the feed industry. And that also will be very contaminated, it's guaranteed. And now I'm reading that wheat, we've engineered wheat 15 years ago, but not grown, not marketed, not commercialized it in Canada. But now I'm reading that there's a push to market it in Canada, which is absolutely ridiculous, because 70% of our crop of wheat goes out to the Asian and European market. And Canada is a huge grower of wheat. And we would lose those markets. The next problem in the environment is genetic pollution. And genetic pollution is about lateral gene transfer. It's a little bit like gene flow, but it's a little bit more urgent, more concerning. The bacterial genes in the plant, in the engineered plants, can move from the plant when the plant is decomposing in the soil. Those bacterial genes can be picked up by bacteria in the soil. That's what bacteria do. They exchange genes all the time. These bacteria can move around in the water, can move to the next stream, to the next river, can move around. And so suddenly you have those genes moving around in the environment. And that can be very serious. The bacterial transgenes can spread from transgenic crops to soil bacteria. And on top of that, those bacterial transgenes can also move into your own body and go into the bacteria that are in your body. We all have an organ here. It's called the microbiome. There's about 100 billion bacteria in your intestine, and they are incredibly important to your health. They are responsible for your happiness. They make 90% of the serotonin that is in your brain. There's a brain-gut connection. That's very important. They make enzymes, they make vitamins, they make essential amino acids. They do all kinds of things for our own well-being. And they don't know that we're human beings. They think they're home. 90% of your cells are not human. 90% of your cells are bacterial cells. 10% of the cells of your body are human cells. That's how important this organ is. So now we're discussing transgenes, from genetically engineered plants, moving on to the plants, to the food, into the bacteria in your gut. That can be very, very important. Think antibiotic resistance. If an antibiotic resistance gene went into your gut, you become an antibiotic resistant factory. Or a pesticide factory if you have a Bt gene going into your gut. In China, we don't do this kind of survey in North America, but in China, they did a survey, they surveyed their rivers, they looked for antibiotic resistant bacteria. And they found antibiotic resistant bacteria in every river they sampled. And the antibiotic resistance gene was a synthetic gene, the blood gene. It came from a lab, and it probably, this is not proven, it probably came from the local engineered crops. And these antibiotic resistance genes found their way into the bacteria in the soil and into the river and into the environment. And there's probably no reason why it's not happening here in North America. This is of very much concern. If there were doctors in the audience, they would like shake their head, you know. Antibiotic resistance is becoming a serious problem in the hospitals, in the healthcare system. And today, and you've been told that it's because we are using too much antibiotic, but the bacteria are becoming resistant. Well, the truth is that 80% of all the antibiotics we use today are used in agriculture to feed animals, because the way we feed them, the way we raise them, is so stressful that they need antibiotic every day to survive. Otherwise, they get sick very quickly. So it's not about using less antibiotics. That agriculture is a serious problem. And now we have this on top of that. It's a threat. Well, at least they are perfectly safe to eat. No. In the early 1990s, the biotech industry came up with this concept called substantial equivalence. And substantial equivalence means that basically the plants are exactly the same. And why should we need to test anything? Because they look like corn, they taste like corn, they grow like corn, they must be completely similar to the non-engineered plants. Oh yes, yesterday I was at the patent office and I said, well, I have this new corn variety and it has a new gene and a new protein and it's unlike any other corn variety on the planet. It's completely new. Yes, thank you for the patent. And then the regulatory agency says, well, I have this corn variety and it's completely similar to all the others. It's like corn, it tastes like corn, this is substantial equivalence. And because those engineered crops are substantially equivalent, they have not been tested ever by the regulatory agencies. They have been commercialized and they have not been tested. What we did not know in 1996 is that the engineering process itself, remember the gene gun, the big gun? Well, shooting pellets into the genome. Well, there is collateral damage, and we didn't know that 20 years ago. We learned that 10, 11 years ago with the end of the Human Genome Project. The Human Genome Project was to basically let us read the whole genome, all the letters of the genome, so we could say, okay, this gene is for this gene is for that. Now we know all the genes, we're going to be able to cure all the diseases. Well, of course it's not happening. And it's not happening because we discovered that the genome is not at all what we thought it was. We thought it was a linear thing. There was about 5% of the genome were coding regions for the genes, and then 95% of the genome was completely weird sequences. We don't know what they're for. They don't seem to be doing anything. It's junk DNA. This is what I learned when I was a graduate student. 95% of the genome is junk. And what we discovered 11 years ago at the completion of the genome project is that actually the genome is an incredibly sophisticated and sensitive ecosystem of genes that collaborate with each other to create proteins. We function with 100,000 proteins, and the Human Genome Project showed us that we only have 20,000 genes. So it's not the one gene, one protein hypothesis anymore. The genes are collaborating with each other and playing with each other and the environment to decide what needs to be done in real time. And there's a lot of feedback loops and a lot of regulatory sequences. So basically, when you come in there and you shoot millions of pellets into the genome, and you blast those gene constructs that you've coated the pellets with, the bacterial genes find themselves in a completely foreign environment, somewhere where they absolutely don't have a clue. And David Suzuki goes like this, he says, where the hell are we? Because it's a completely different environment, and the regulatory sequences are completely different. And you should expect that new proteins will be created. And this is what we see. New, unknown, rogue proteins are created by this technology. And this we didn't know 25 years ago, 20 years ago, and the regulatory agencies and the biotech industry is completely ignoring it. In Europe, they know that. We have studies to show that there are new proteins created in those crops. Engineered corn, soybean, and canola all have variant, new, rogue proteins, different from what the biotech industry reports in the engineered plants. And there's a good number of those publications showing that the proteins are disrupted, are truncated, are misformed, are poorly shaped. And they're all enzymes, and they all have, many of them will not do anything, but some of them will actually be active, and we don't know what they're going to do. That could be dangerous. In 1996, just before the crops were commercialized, the toxicologists, the experts of the Food and Drug Administration in Washington, D.C., warned their administration that there would be serious problem with the technology, that there would be new proteins created, and that some of those could be toxic or allergenic or cause nutrient deficiencies, etc. And they warned their director that those crops should go through the whole testing regime of the administration. And their director completely ignored them and signed up to Monsanto, and the crops were commercialized without any testing. Their director was Mr. Michael Taylor, and he had been a lawyer for Monsanto, and now he was in place, head of the FDA, had been put in place by the White House, and then after that he became vice president of Monsanto. And today he's back at Monsanto. He was away, and then now he's back at the FDA, to make sure that the corporation is well taken care of. So, outside of North America, because in North America we live in a corporate bubble, I call it a bubble, the corporate science community that works basically for the corporation, generate a lot of studies showing that the technology is perfectly innocuous and everything is safe, and no, there are no new proteins and there is no evidence of harm. And they will be very happy to let you know that there are hundreds of studies, of safety studies that have been done that show absolutely no harm. Except outside of North America, there are also a lot of studies that have been done and that show serious concern. A lot of organ damage in rats. These are all animal studies, lab animal studies. So we have allergies, severe allergies in rats, immune response, anaphylactic shock, leukemia, and toxicity, toxic protein that cause organ damage. This is mice-fed engineered soy have damaged liver. Mice-fed engineered soy have damaged testicle, uterus, and ovaries. This is mostly out of Europe. And then there's 2 more studies out of Europe. I'm not going to show you 100, but this one is interesting because it's, I was almost there. I was in Edinburgh in 1998, in August 1998, for a week for a conference. And during the conference, there was a scientist that was working in Aberdeen in Scotland, who had been researching these engineered crops. He had had a large grant from the European Union to put together a protocol of research to test engineered crops, with animals, to make sure that he was not going to get any false positive or any false negative. He was one of the top toxicologists, and he was asked to do that, and he did, and he set up experiments with feeding rats engineered potatoes. And after 4 months, he decided to stop the experiment because the animals were really severely damaged. And he went to his director and, with his agreement, stopped the experiment and decided to go public. There was no doubt in his mind that there was something wrong with the technology that was causing organ damage in the rats. He went public on TV, that's when I was there, and apparently there were phone calls between Washington and London, and to London to Aberdeen, and he was fired the next day. And he had a gag order, and his papers were confiscated, and his lab was closed, and his research team disbanded. And this was one of the top experts in the world. And there was such a scandal in the scientific community that after a few months he was given his papers back, and he quickly published his results in one of the best medical journals, one of the most prestigious medical journals in the world, The Lancet, showing organ damage. This is the lining of the stomach of the rats. Precancerous, he calls it. And the last study I'm going to talk about is where I did my undergrad in France, in Caen, in Normandy. There is a professor there that did a study for 2 years. When Monsanto wanted to commercialize their crops in France, they were told to do a study, a feeding study with rats. So they did a feeding study with rats. They picked the rat strain, and did a protocol, and they fed the rats for 3 months, and there was no signs of any toxicity of any kind. And so they showed their data to the regulatory agencies, and that was that, and the regulatory agency accepted that. And this professor was intrigued because he had done some work, and he knew it shouldn't be that way. So he asked Monsanto, can I see the data? And they said, oh, absolutely not, this is top secret. And so, because it's not North America, it's France, he went to court and did win, and he was allowed to see the numbers, the data, the statistical analysis of the results of the study. And he wasn't happy with the statistical analysis, so we had a team of statisticians, and they redid the analysis, and they showed statistical differences, significant differences between the treatments, which, if you're a scientist, means that there is definitely some clue as something going on. And he showed that to Monsanto and the regulatory agencies, and the answer was, oh, yeah, well, that's not biologically relevant. He was not happy with the answer, so he decided to repeat the experiment himself. He chose the same strain of rat, the same protocol of research, same everything, except he decided to look at more parameters, and he decided that instead of 3 months, he was going to extend the length of the study for 2 years, the whole length of the life of a rat. Well, after 3 months, there wasn't much happening. After 4 months, there was organ damage. This was a toxicity study. It's the same as Monsanto set up. It was to show toxicity. So there was organ damage after 4 months. And then a few more months, well, the rats started dying, and most of them started developing tumors, breast cancer. And he let, you know, the previous researcher had decided that that was enough, but he let the study run for 2 years, and the tumors got bigger and bigger, and more rats died for 2 years. This particular study was published a year ago, and you wouldn't believe the level of noise that the biotech industry and the corporate scientific community has done about this one. This has been completely criticized, debunked, that it's not acceptable, it's not acceptable. Even the regulatory agency, the government in Canada says, oh yeah, no, this is not good enough, this is not good. This gentleman, this professor from France, has answered, first of all, it was exactly the same experiment. He was criticized because he chose the wrong strain of rat. He was criticized because he didn't have enough replication to do a cancer study, but he wasn't doing a cancer study. He was doing exactly what Monsanto had done, a toxicity study. He just happened to stumble onto cancer. Anyway, this study is being repeated as we speak. So that's the end of my talk about genetic engineering. So I've basically gone through all the promises of the industry showing you that it's not safe to eat, at least for rats. And it definitely has an effect on the environment. And it doesn't increase yield. And it certainly does not decrease pesticide use. Okay, a few more minutes. Now it's about Roundup. Because Roundup was invented in 1964 as a chelating agent. This is glycine, methyl, phosphonate, all the salts of phosphonic acids are chelator. A chelator is a molecule that grabs on to metals, okay? If you all know that you need iron in your blood for hemoglobin, because there's a metal ion of iron in the center of the hemoglobin molecule so it can do its work. That's why you need iron. If you don't have enough iron in your blood, your cells are not breathing properly. The hemoglobin does not transport oxygen properly. It needs iron. Most of our enzymes need metals to function. So a chelating agent is very useful to clean up water or to clean up industrial processes, but it's not necessarily useful in your food. And remember, 90% of all engineered plants are sprayed with Roundup. That's by definition. Therefore, you have Roundup residues in 90% of all engineered ingredients. It impairs many enzymes. It's a very strong chelating agent. It was then discovered to be a broad-spectrum herbicide. That's when Monsanto bought the patent for a herbicide. And then they also patented it as an antibiotic. Roundup is an antibiotic in your food. It's very toxic to fish. It's very toxic to amphibians. We've known that for a long time. It's teratogenic, causes birth defects. It's also very toxic to bacteria, obviously, because it is antibiotic after all. This study, published in Clinical Pharmacology and Toxicology, it's the study of the effect of glyphosate, the active ingredient of Roundup, on enzymes on liver and intestinal activity in the rat. And that study discovered that this enzyme, this chemical, glyphosate, inhibits a family of enzymes called the cytochrome P450. And cytochrome P450 are very essential to very basic processes in our cells, and in bacterial cells, and in most living organisms. And we have seen from other studies that in the rats fed engineered grain laced with Roundup, we see gastrointestinal disorders, kidney and liver damage, and infertility. When I say gastrointestinal disorders, I don't mean tummy ache. I mean autoimmune disease. I mean celiac and Crohn and leaky guts. Very serious stuff. And allergies. There's two researchers from the Massachusetts Institute of Technology who published a report last year because one of them had been studying for a long time autism in children. She's a brilliant biochemist. And she's been very intrigued that 20 years ago, there was one case of autism in 10,000 kids. And today, it's 1 in 50. 1 in 50. And she says, there has to be an environmental factor. It has to be in the air, in the water, or in the food. And so when she saw this paper, this study, she went back to look into the literature to see what would happen if you inhibit this family of enzymes. And what would happen in humans, that is, would be exactly what we saw in the rats. Gastrointestinal disorders, kidney and liver damage, infertility, obesity, depression, autism, Alzheimer, and there's a few other, and cancer. And the biotech industry is very quick to say, and I've heard it many times, but millions of people have eaten trillions of engineered meals, of engineered food, trillions of meals with engineered ingredients, and nobody has ever been hurt. And this is, you know, I used to think like, well, I don't know what to say to that. I mean, it's true. How do you know? But not anymore. I'm saying now, this is a completely empty statement. There's no study. There's absolutely no backing up this statement. There are no studies, there is no data, there is no research to show that nobody has ever been hurt. There is no acute toxicity to Roundup. You don't die in half an hour from Roundup residues because it's really quite safe, the industry says. But it does inhibit enzymes, and it's a very slow and insidious process, and it causes all this damage. What we have are correlation studies, correlation curves, showing that really there's a lot of people that have been admitted in the hospital with kidney damage or liver damage or obesity or cancer or all these diseases that I just mentioned. And the curves are basically, you know, kind of basic at the bottom, and then starting in the mid-1990s, they start going up very quickly when glyphosate became prevalent. And we have curves like this for celiac. We have curves for, this is for kidney injury, and I could show you curves for all those degenerative diseases that I mentioned. And that's what we have, nothing else, because there's been no research done on humans, because the industry says it's perfectly safe, and some people say, well, maybe we need more research. I say the research is in, the data are in, and we know. I'll just show you two or three more slides here, because a lot of people often ask me, well, what can we do? What can I do? And Dr. Chopra will have a different version of this, but in terms of the research that I presented, what you can do is basically remember that Roundup is absolutely in all engineered food. Engineered ingredients are laced with Roundup, which is antibiotic, which is not really good for the microbiome here. Actually, it's like basically the, you know, a comparison would be like, think of the cattle in those big operations where you got 10,000 heads of cattle, or the chicken, 20,000 chickens in a barn, fed low-level antibiotics every day. That's where we are. So if you're in avoid industrial food, prepared food, processed food, junk food, canned food, baked food, anything that has been prepared and processed. Contains corn and soy and canola and sugar. If you go into the store, and of course organic is your best bet because 1%, there's only 1% level of contamination that's allowed. So you're sure if you buy organic that it's 99% without GMOs. If you're in the store today, the vegetables are sprayed with insecticide and fungicide perhaps, but at least they're not engineered. Only sweet corn is engineered. Sweet corn is engineered. You want to know that. If you look at the fruit, none of the fruits are engineered, except papaya. If you looked at the meat department, unless it's organic, the cattle, the chicken, the pork, they've all been fed engineered grain with Roundup. And if you look at the rest of the store, there's a lot of engineered food there. That's it. So organic is good. Local, fresh, grow your own. And one thing that Dr. Chopra and I are really insistent on is that we are the messengers. We're here to let you know that this is happening, and it's up to you to make your voice heard. So talk to your friends, talk to your family, talk to your neighbors, everybody, the grocery store manager, let them know your concern. Talk to your MLAs, your MPs, your municipal council. Write letters. Make sure to let everybody know that you are really upset. And if you're really upset, go for it. There's one publication, if you're curious and you want to read a little bit more, this is available on the internet for free. It's a compilation of studies done in Europe. It's called GMO Myth and Truth. It's available at this website, earthopensource.org. You can download it for free. It's very easy to read because it was compiled, put together by a genetic engineer, but summarized by a science writer who does a really good job to make it really available, easily accessible. You don't need a PhD to be able to read this. Thank you.